I'd like to introduce our next presenter. Her name is Beth. She earned her PhD in molecular immunology and biochemistry from the University of Maine and continued research into inflammation and lymphocyte development as a postdoc at both the University of Maine and the Maine Medical Research Institute. Beth has over 20 years of experience in flow cytometry and research methods and analysis. She joined Verity Software House as an application specialist over 8 years ago and has since enjoyed providing customers with solutions for their high-dimensional data analyses. So with that, let me toggle the controls over to you, Beth.
Thank you, Janelle.
Here they come.
All right. Okay, so you should be able to see my title page and hear me.
Yep.
Okay, thank you.
Everything looks great.
Great, thank you, Janelle. Thanks everybody for joining us. So we understand that there are so many different analysis programs and algorithms to choose from, and there's also a great deal of confusion about the best choice for analyzing high-dimensional data. So we would like to offer you a solution that can not only analyze your data, but give you a whole different way of looking at it. GemStone has a revised user interface and added features that provides you with a comprehensive package that will help you get the most out of your data. GemStone features a multifaceted approach to data analysis. So step-by-step wizards will help guide you through building a template model using your panels and your experimental system. Templates can then be used for any number of data files using the automated batch system. And there is no limit to the number of measurements. So more measurements are actually better as it provides the program with more information to work with. Data cleanup can be automated and based on objective equations. And once a model is built, it takes only just minutes to analyze a data file, and that includes the Sensei Graph calculation. Multiple users of that template will get the same results, which makes it a very effective tool to share with your collaborators. The driving force behind GemStone is called probability state modeling, which is a published, patented, well-tested algorithm that eliminates the need for subjective gating. Sensei, which is GemStone's high-resolution dimensionality reduction algorithm, is calculated independently of the model in an unsupervised manner. Sensei automatically labels the cell type populations and subsets, as well as providing a consistent orientation from sample file to sample file. All cell types are clearly defined with subsets and subpopulations of interest. Unique overlay plots display correlated measurements and provide a clear measure of comparison from sample to sample. Reports are constructed automatically and are fully customizable and editable. All graphics are also saved automatically during the analysis process in your choice of format. A full database is calculated and exported for further analysis. And you can also set up smaller tables on the report and populate them with custom equations, alerts, QC metrics, etc. Tricon graphs, which are not shown here, are a powerful tool for enumerating rare populations, unknowns, activation markers, and cytokines, whether singly or in any combination. There are built-in quality control measures, which include stain assessment, which uses the strictly standardized mean difference method to evaluate staining. There's a contingency table that evaluates the overall model performance and reduced chi-square values that evaluate modeled cell types. Alerts, flags, and equations can be set up to identify unexpected or out-of-range results. So all of these features and more actually combine to provide a comprehensive automated analysis that is interpretable, reliable, fast, and reproducible. So now I'm going to focus in on just a few of these features, and instead of telling you about this, I'm actually going to show you how and why this works. The driving force, as I mentioned earlier, behind GemStone is called probability state modeling. The original GemStone was released in 2009, and the paper describing the algorithm published here in 2015. So GemStone is not a black box. You can see your entire analysis, you can make changes, you can confirm for yourself that things are working as they should. There is no scripting or coding involved. So what is a model? A model is comprised of one or more cell types, such as CD4 or 8 positive T cells, B cells, etc. But these cell types are based on your panel and your experimental system. Within the cell types are expression profiles that control the fitting of the measurements in the panel. The expression profiles determine the probability of an event being included in a cell type. And they do this with what we call control definition points, which I'll actually explain shortly. So what is an expression profile? So an expression profile is actually a different way of looking at a 1P histogram. On the top right here is a very simple example of a DNA measurement with the typical intensity on the x-axis and frequency on the y-axis. If we take that histogram and turn it on its side, we now have frequency on the x-axis and intensity on the y-axis. To become an expression profile, we even out the events, so all events are distributed evenly over the expression profile. The x-axis becomes cumulative percent, which shows 0 to 100% of the events. The y-axis, again, is intensity. There is a peak finder system within the program that identifies the peaks based on the density of events. And you can see this little black triangle here on the right that shows you where that peak would be. We call this particular profile is called a constant profile. And there are some cell types that will only use constant profiles for enumeration. At this point in the modeling, this is when a raw data file is added, the events are actually in acquisition order, so you can see if there are any problems like clogs or bubbles during that acquisition. We can also model progressions. So if we try to do this with gating, we have the problem of subjectivity, double counting. We lose the ability to really look at those transitional events, and it's very difficult to scale up the analysis. The histogram shown here is CD27, which is expressed in this case in a peripheral blood B cell type. There's a negative peak here in blue and then a positive peak here in red. We can, to demonstrate modeling of a progression, we'll start again with that same CD27 histogram. If we turn that on its side, again, we have a negative population here in blue, the positive population in red. The x-axis is frequency and the y-axis is intensity. To become an expression profile, again, the events are distributed evenly along the expression profile. The x-axis is called cumulative percent. Progression here, it's percent of events. We can see the black triangles, which identify the negative and the positive population for CD27. So this particular sample is a B cell example, and we know that CD27 upregulates when B cells progress into memory B cells. We can see again that it's two black triangles which identify the negative and positive. The program actually positions the CDPs, the control definition points, based on those peaks for what we're going to call a step up, because CD27 upregulates. When modeled, the program actually rearranges the events so that the CD27 negative events are to the left here in blue, and the CD27 positive events are to the right here in red. We account for the overlapping transitional events between those two populations. The x-axis here shows us that approximately 80% of the events are CD27 negative, and approximately 27% of the events, 20% of the events are CD27 positive. The x-axis then becomes a common axis. If we look at this in an actual cell type, the common axis actually allows for scalability. In a cell type, the expression profiles are stacked, and each event will be evaluated and assigned a position. Every event in the sample file is accounted for. If I drew a line down the stack, we can see that the same event would be in the same position in each one of these expression profiles because all of these markers are correlated. This then becomes the basis of the overlay progression plot where you can visualize how the markers change and interact. In this case, it's B cells. We see CD27 here. It upregulates into the memory B cell population. These control definition points, for each expression profile, there's at least one set of these control definition points that define how that expression profile will fit the measurement data. So this is shown right here, and a little blow up right here for you. So each of these has 3 parameters that are associated with it. We have intensity, which is the Y position. We have a state, which is the X position. We have a standard deviation, which is also called line spread. It's showing us the 95% confidence limits. These CDPs are mathematical functions that actually define a probability distribution, and we will teach the model to select for that peak of interest by positioning the CDP on that peak of interest. The CDPs are flexible so that they account for variations in intensity, but they're constrained by what we call an estimation range to only select that peak of interest during the fitting process. And during the analysis, the program will calculate a probability for each event using all the information from all the cell types and all the expression profiles as well, and then calculate a total probability for the event, which will determine where the event is assigned. So again, every event in the data file is accounted for, and any unassigned or unclassified events are enumerated. So now I'm going to switch to GemStone and give you a demo of the wizards and the modeling process. So instead of telling you, I'm actually going to show you. And we will also take a quick look at the results and the analysis output. I'm going to bring up one of my GemStone programs. So when you open GemStone, you have a nice blank slate here to get started with. And if we wanted to create something called a project, we would click the New button, and we would be able to name our project. We could base it on a previous project that we collected. But I've also already set up a project, and I've just named this webinar demo. First thing I'm going to do is add my data files. When I click that Add button, it brings me to a navigation window where I can navigate directly to my project, or I can navigate out of that into a place where the data files might be stored. I'm going to open my folder here called Exported. And these are the same 5 files that we've been looking at. These have been previously cleaned with a model that I've built and done, and cleaned the data files automatically by removing the debris, removing the aggregates, and the dead cells. So I'll multiple select, I'll add them to my batch file. So we have 5 files now in our batch. To develop a model, I'm going to click this Develop button, which automatically opens up a tab that has all of the tools that I need to build a model. First thing I'm going to do is I will just select a file. I'll read in the file. And we'll know that the file is read in. If we look here at the bottom right-hand corner, we see the name of the file, and we also see the number of events. The next thing I'm going to do, if I were going to build a model from scratch, I'd go to my measurement wizard and I would be able to look at all my different measurements. I would be able to adjust my scaling. I could edit the measurements if they weren't named correctly. I could also check my transforms and make sure that everything was on scale and transformed properly. But for the essence of time, I'm just going to skip right to my cell type wizard. I'm going to open that. And I have my different options in my cell type wizard. I can create a new cell type. I can create a report page. I could also edit the cell type if I had a template already built. Today, we're going to just create a very simple cell type. I'm going to create a CD4 positive T cell. I'm going to leave it as a competitive cell type, and I'll go to my next. Now, I'm going to talk a little bit about selection, and selection markers are really those iconic cell type lineage markers that you would want to choose for your B cells, maybe CD19. For T cells, we're going to do CD3 because we know T cells are CD3 positive. I'm going to just pick CD4 and CD8, very simple selection markers. I'm going to click next. For my staging markers, we're going to talk about, this is really where you're going to define your cell type subsets. So if I'm looking at T cells, I might want to look at different subsets such as my naive, my central memory, my effector memory, or my effector subsets. I'm going to make this very simple today. I'm just going to choose CD27. I'm going to choose CCR7. I'll click next. And functional markers would be unknowns like cytokines or activation markers. I'm choosing just one for a simple example, and I'm going to choose PD1. And I'm going to go to next. So this is really where the fun starts, and we're going to actually build a model. Now we know that CD3 for T cells are positive. We see two distributions of events here on the right. We have two different peaks. So this would be the CD3 negative peak, and this would be the CD3 positive peak. Our CDPs come up on this positive peak. We see the line spread, 95% confidence limits. This is called the brightest peak. We also have some other choices here if we have different peaks that we want to identify. Right now, I'm going to leave it as it is because I want CD3 positive, and I'm going to hit preview. Preview actually goes through the data file, will identify the CD3 positive events and select those and fit that expression profile. So now we've actually selected all of the CD3 positive events in that sample file, and we can see right now there's 120,000 events. I'm going to click next, and which is CD4. Again, we want CD4 positive events. This is a normal distribution. I'll click preview, and now we've selected for CD3 positive, CD4 positive events. And you can see the number of events has changed to about 64,000. I'll go to my next profile, and this is CD8. This would be what we would call a negative selection marker. And what we want to do here is just sort of clean up the CD4 cell type by eliminating these dim to positive CD8 events. I'm going to change this to my dimmest peak. I'm going to do something called a low pass distribution. We want a low pass because it will grab all of the events from here below. So if we have any kind of unmixing or compensation issues, we're still selecting all those events that may be below 0 or maybe below the other negative events. I'll click preview, and then you can see it's cleaned that up. I'm going to go to our next profile. So here we'll do something a little different. For T cells, we know that CD27 downregulates as T cells become antigen experienced. So we call this in GemStone, we call this a step down. I have two peaks, and I have CDPs that are positioned on either one, on both of those different peaks. There's two sets that are linked. I'm going to shut off the width from peak, because what I want to do is I want to change that line spread to gather all of the, to consider all of those transitional events and to account for all the transitional events. And then I'll click preview. Right now the program is looking for all those CD3 positive events, CD4 positive events, CD8 negative events, and it's fit the expression profiles for CD27. It's rearranged the events so that the CD27 positive events are now to the left. The CD27 negative events are now to the right. And you can see this as a step down. If I go to my next profile, which is CCR7, you can actually see the data start to have some structure. It automatically will determine that you want to step down. I'm going to do this again. I'm going to just change that width. To 6. We count for those transitional events. I've got to go to next profile. Sorry, I should have just hit preview, but it actually I could probably go back, and I'm just going to show you. I don't know, maybe I'm not going to. I'll hit preview here. Okay, so we got back to the same place. So now all the CCR7 positive events are to the left, CCR7 negative events are to the right. And it's that difference between where CCR7 and CD27 downregulate that will form the staging. I'm going to go to my next profile. Here's one of my functional markers. I'll hit preview. This is what we call an unclassified marker. So what we're going to do with these is we're interested in these positive events for PD1. We will eventually put those into a tricom to enumerate those events, either singly or with other markers. So I click summary. I have a couple of choices here. I can save my template model, update the summary map, add to my Sensei, add database fields directly to the database, and I'll click finish. So you can see here that a report page has automatically been created. These are live. You can add any kind of dot plots. These dot plots are really here just for comfort so that you can see your data in a familiar way. We have some tables that are already created. This is a table of intensities, a table of statistics. Phenotype is here, and you can add more. You could add some, subsets or add additional pages. If we were going to build a model, a complete model, we would continue this process and go through each one of our cell types, adding CD8 T cells, B cells, monocytes, NK T cells, etc. We have a summary map which shows us what's been modeled. And then I'm going to come into my cell types. Each one of the cell types in our data file will have a workspace. The staging is calculated automatically. You can see that CD27 downregulates, CCR7 downregulates, and this again becomes the basis of that overlay plot. We can follow this over for CCR7 downregulation, CD27 downregulation, and the difference between these two form that staging. So for the sake of time, I'm going to just bring up another program. So this is a completed data file. I have done all of my subsets. You can see that there's a report set up with a summary table of statistics, QC table. I have my Sensei that's been calculated automatically. I have a report page for each one of my cell types. This is what a stain assessment page looked like. We have metrics that tell you what your stain, how well your staining actually is. I have a summary map with our unclassified events or unassigned events evaluated. I have all of my cell types. I have CD8, CD4, NKT, T star gamma deltas, B cells, NKs, etc., monos, MDCs, PDCs, and monos. I'm going to go back to my Sensei and just show you that this is actually interactive. I'm going to just bring up my edit window. I'm going to bring it over here to that. The populations here are automatically labeled to you. Again, this is calculated independently of the model, but it's linked into the model so that all of the populations are actually identified. This is, we can look at frequency, heat map, so you can look at where the populations are, and then we can look at intensity heat map, go through each one of our markers and scroll through, see what the expression is. The other thing that we can do is draw a little region around a population that might be unclassified or unlabeled. We can animate this. We can go back into the cell types and look where those events might be animated, and then we can actually scroll down through and look down at the phenotype for that. And it works both ways. So it's a process that is a two-way process. We could go into any cell type and do a lot of different animation. So the orientation of these senseis is the same. I'm going to bring up one more thing here. The project, everything is saved automatically in the project. It's there's a project library. A project consists of all these folders. I can come into my graphics. These have all been saved automatically. I can look at my different senseis. You can see that the orientation, the CD4 T cells are all oriented up here in the top right. And we can see this data file here that has a very different looking population. We also have PDFs that are saved out. We have different, all the graphics, a report. I'm going to come back here to my PowerPoint. Maybe. Okay, from this slide. Sorry, get a little bit of a lag here. Okay, but this is the Sensei Graph, which is looking at two of these donors. So we have this really pretty normal looking population here of CD4 cells, and then we have this other population that has kind of been brought out. So what happens when you have something unexpected like this? We can see that this is a very different population than the CD4s, but it is somewhat related. It appears to be related to that sort of terminal effector population, but it's separated by the CD4 T cell. We can look at this in a different way. Also, we can look at the overlay plots. We can see this greatly expanded effector memory or effector population. And we can see that CD57 is expressed here, CD56 is expressed, and PD1 is also expressed. You can look at heat map intensities to see the differences between data files. CD3, very bright for CD4, CD56 is expressed, CD57, and PD1. So in summary, I've shown you a number of different features in GemStone. These combine to give you a fast, automated, objective, interpretable, and reproducible analysis for any number of measurements or sample files. So importantly, GemStone also provides you a different way of looking at your data with these overlay plots, summary maps, Sensei, etc. So I don't have time to tell you more today. The project format makes collaborations very easy. You can share the whole project with your collaborators. Your analysis is secure and at your fingertips. And thank you very much. And thank you, Cytek, for the opportunity to talk today.
Thank you, Beth, for your presentation. I think we've got time for a couple questions before we transition to our last speaker. One is, what if you don't know the biology of all the different markers in the panel you're looking at, and you're trying to look for some novel subsets? What would you recommend for those types of users, those types of applications?
This question comes up a lot, and there is a reason that you develop your panel in the first place. So you have some markers that you are interested in. You have an idea about what your what some of the cell types would be. So, you know, depending on your experimental system and your either tissue, your blood samples, you have certain markers that you've chosen for your lineage markers, your sort of iconic markers, and then some of those unknowns. So what we really do in GemStone is we model what we know. So we know, you know, you really only have to model a few markers of what you know. And then it will very clearly show you what you don't know. And you have the opportunity to do that sort of exploration with GemStone. You have QC metrics that will help you. If you make a mistake, it becomes very obvious. We've done a lot of different cell types at a lot of different tissues at a lot of different samples, so we can always help you at Verity too. And, you know, we try not to encourage phishing experiments because I think, you know, most people don't, but but we know we all do that. So there are different ways to do it, and we do have ways that we can help you do that.
Thanks, Beth. I'm going to take one more and then we'll transition onward. Can you use the progression analysis to compare two samples? For example, if you have T cells that are treated with PBS versus T cells that have been treated with some kind of drug.
Absolutely. It's a really powerful way of looking at, because when we model things like leukemia, lymphoma with MRD, you know, those differences become extremely obvious. That the staging often is what changes. So it could be expanded. You might have a very expanded, if you're looking at T cells, a very expanded naive stage. If you're looking at, depending on what you're looking at, you can really see the differences between data files. Yes.
Great. Thank you, Beth.